right?
Okay.
Yeah, try to be close here so there's some people.
Yes. Okay, I got this habit, brought the acknowledgements at the beginning of the presentation because it's very important to tell you that this is not my work exclusively, but of a large team. Let's see this one. 1868, the University of California is established based on the Morrill Act signed into law by President Lincoln. And then 1870, this gentleman, Saunders, working in Washington, D.C., received 12 navel trees from Bahia, Brazil, which they have found their way from North Africa. The Arabs brought the citrus with them, North Africa, into Portugal. And then the Portuguese colonized Brazil. Then he grew them, he repropagated them. 1873, he sent 3 of them with a train to San Francisco, and then with horses to Gilroy, and then a stagecoach to Los Angeles, and then the Tibbetts family 65 miles to Los Angeles to pick up their trees. Okay, 5 years later, or actually 6 years later, the parent navel tree won in competition, and the commercial interest began. Okay. So with this commercial interest, this gentleman, just as a representative story, John Reed, and of course the whole industry start asking for how do we grow this thing? And that's true not only for citrus, but in general, people were asking the university, teach us how to do things. So late 1800s, the extension services come into place. And in the early 1900s, we get the Citrus Experiment Station in Riverside, and that's our afternoon visit. The CCPP operates at the original 1907 Citrus Experiment Station. And now you have a group of dedicated people doing research, citrus research exclusively, right? A nice group of people, horticulturists, entomologists, breeders, pathologists, like the father of citrus virology, Dr. Fawcett up there, that he made a pivotal discovery in the 1930s. If you go to a sick tree and you take budwood and you make trees, those trees will also be sick. Sounds very trivial today, but it was a major breakthrough. And based on that discovery, you Californians, or in the audience, whoever are Californians, you have been selecting at the citrus nursery with the help of the state, of the university, of your industry and division. Since 1937, you have been selecting against diseases. Very, very important. And you haven't stopped since then. 1941, Dr. Wallace was extracted from USDA from Dr. Fawcett. He visited with him several times. The daughter of Dr. Wallace shared with us his journal. So he writes down, Dr. Fawcett came, he spoke about these different things, and his salary, his salary would be $3,800 a year. And he decided to leave USDA and be one of the first people to start exclusively working on citrus virus. The citrus virology, remember, started like less than 10 years earlier as a field of science. And with Wallace working at the experiment station, discoveries keep piling up. Now we have more tests. We have biological indexing developing, and we're focusing on several diseases. And now we are almost 20 years later, and the world is catching up, and they come together in 1957 in Riverside, and they have the first conference on citrus virus disease, and they realized this is going to be a big deal, and they formed the International Organization of Citrus Virologists. And we have a continuous service and collaboration around the world. The IOCV is still active. We published many books, and now we have the Journal of Citrus Pathology available online for all of you. Same year, 1957, when this whole global thing comes together, the Citrus Advisory Board asks the University of California to start a clean program for the state. And that's the beginning of the CCPP. Another major discovery and change in 1957, I'm going to spend a little bit more time in this because it really transformed the nursery production, not only of citrus, but globally for all the crops. The UC system to produce plants in containers. is published. And the concept of having artificial potting mixes is not new, goes back to the ancient civilizations. But this gentleman is credited for spending sometimes from UC Los Angeles. Philip Chandler spent some time in John Innan's Institute in England and brought back the idea of having these things, plants growing in artificial media. He writes in his publications that the raw materials that the English had access, we didn't have in California. So there was a lot of research. What do we have in our hands in California to help us make the UC mix? And they came up with the peat moss and sand. And then later in 1967, research at CCPP adjusted the original UC potting mix recipe exclusively for citrus. And there is this beautiful diagram here. I don't know if I can move my mouse. And it may have changed today what you do in the nurseries, but the principles are the same. We may have replaced peat moss with coconut coir, but all of us add micronutrients and macronutrients and so on. And you can see and of course, it's not only the potting media, but sanitation, right? Steaming the pots, cleaning the benches. And that's a picture from the original paper, how much growth you go on a soil used at the local nursery versus the UC mix. And from, again, talking with old-timers, here is a nice story from this gentleman sanitizing the CCPP benches there with copper naphthenate, Mr. Reginald Wagner. After the CCPP was done emptying the pots, he would take the soil that was to be trashed, take it home, and his hobby was to grow chrysanthemums. So he would enter local, like in Pasadena, he writes in his notes, enter competitions back to back, and he just decimated the competition because nobody was growing at that time flowers in artificial potting media, right? They were digging soil from in a hole in their backyard and doing this. And like he won 90% of the awards one year, 70 the next, and the third did the same thing. So everybody hated them, but but they draw the attention, right? When you have these kinds of results. So the world follow up with this, and we switch to putting controlled media in our pots. Major contribution, and we keep building on it. The next slides will be like building on that a little bit. Also in the 50s, the federal government caught up and they gave us the regulatory frame in citrus nursery production and citrus movement of germplasm. It's prohibited from entering the United States unless under permit. And also in the 50s, we started the experiments with heat therapy for removing pathogens from stock. And we start the foundation block in Lincoln, which still exists in the case of California, but also something I didn't know. They had multiple foundation blocks those days in different, like in Ventura and in the desert. 1961, Chester Roystacker there from CCPP plants the first budwood source tree at Lincoln. 1963, the first budwood from the clean program arrived to the nurseries. 1968, the Citrus Research Board come together to support, among other, the CCPP and the registration program. In 1969, there is the publication that says you need to use bleach to sanitize your tools. So let's spend a little bit more time on that because this question keeps surfacing. So we're going to together read the table and you spread the word. I don't want to hear this question ever again. Okay? Bleach is the only thing that works. You see those zeros there? That's what you want. Zero transmission. Okay? Here is everything else that doesn't work. Okay, and here is your alcohol right there, alcohol and flame, it doesn't work. And here is Lysol, it doesn't work. And I got a hold of a chemistry professor that works with chlorine and so on, and I told him, okay, tell me what's happening, explain to me exactly. And he said, whenever you have these products, either sodium hypochlorite, which is in household bleach or tablets of bleach. When you add water, they convert to hypochlorous acid. That's the actual chemical that does the job. And he also said that, it does the job because it completely messes up the structure and the chemistry of the bases that make DNA and RNA. So it kills all the pathogens, no matter if they are bacteria, viruses, or viroids. A key element, though, here is that the bleach, it's stable at high pH. The moment you dilute it with water to use it to spray your tools or clean surfaces, pH is lower, so the hypochlorous acid starts breaking down, and that's why we recommend to people to always use a fresh solution of bleach. Also, organic matter starts deactivating the bleach and so on. They even dipped, is it phytotoxic? Is it going to ruin our tools? If you spray your tools with W40, back then they were using a mix of vinegar and stuff, and you wipe your blades clean with a cloth, nothing is going to happen to your plants, nothing is going to happen to your tools. Yes, it will get corroded, but you're avoiding issues. So they dipped 30 buds for 1 second in 20% bleach, and then they propagate them and they grew. Okay, so bleach is working, nothing else is working. Spread the word. In 72, Semantic discovered the smallest citrus pathogen, which is just a piece of 300 nucleotides, more or less, of RNA, the exocortis, and then later on, through innovation in gel electrophoresis, whatever they were calling exocortis, they discovered, you see all these black lines there on the gel, that there's actually multiple species of those viroids. Okay, very, very important discovery as well. And this is like the holy grail. In 72, Professor Muresigi here in Riverside, he said, I think I can recreate citrus varieties with the apical meristem without any diseases. In 75, a group at CCPP modified this technique to the form that we have together, but innovation, and you will see a lot of this afternoon, keeps happening in this field. 77, the program takes it, the name that has today, from it used to be called all these years the Citrus Variety Improvement Program. It's named to Citrus Clonal Protection Program. And the biggest thing that happened in these later dates in the 80s is that the director of the program petitioned the federal government for plant material to come directly to California and not get stuck for 15 or 20 years at the federal quarantine facility. So that was a major breakthrough, and that's how we continue doing business together. And we're getting into the 90s. Because of the threat of Tristeza, we built our first protected foundation block at Lindkov. In 2005, the industry in collaboration with the CCPP, up to this day, a professor of plant pathology or virology would become, it was like a sidekick to be the director of the CCPP. But they realized at that time that they needed a dedicated position, and that's what they created. There were interviews performed in April 2005, and this is something that will be ingrained in your memory forever. This is me the night I finished typing my PhD thesis, and this is me the morning of the interview. And when we have a break later today, I'll tell you exactly what my professor told me, because I cannot use the language in public, what to do with my beard and my hair. So I got the job. I became the director of 2005. The same year, the Nursery Society, no, the society still exists. The Nursery Advisory Board became a marketing order so they can fund research themselves. And in 2009, we have the National Clean Plant Network that was transformative for all of us, for all specialty crops, but especially for citrus. And this is the frame that we're operating today. How am I doing with time? I'm going okay. And again, this is the, so if we can zoom in what we have been doing as CCPP in the modern years, we are a highly collaborative program with state, federal, industry agencies. And we do two things. We make sure that material enters the state and the country in a safe manner. We make sure it doesn't have diseases. And then we distribute that clean material to whoever needs it. The program looks like this, like a pictorial. We have the quarantine part in Riverside, and as soon as the different steps of therapy and testing is telling us that the material doesn't have any pathogens. It crosses the red quarantine line and it moves in the heart of the industry at the Linkov station. And from there, from the foundation facility, finds its way in all different ways, both for research and for commercial production. And then that diagram in a flowchart looks this way, which is essentially a constant loop of therapy and testing until we get negative tests, and if something tests positive, it goes back to therapy. And until we get all negative tests, the material is not moved towards release. As soon as everything tests negative for different diseases and pathogens, it moves towards release from quarantine. So what have we been doing all these years to improve those steps and make sure we're doing a better job to support nursery production and, of course, ultimately citrus production in the United States? This is when I started, this is how many people we had. As I say, me and 3 old guys, and definitely we needed help. So this is how we look today with the support of the nurseries, the CCNB, the CRB, the Citrus Research Board, the growers, the National Clean Plant Network, the university. We have a proper structured program. You will meet a lot of these people. They're already in the room, but also in the afternoon when you visit the CCPP. We got a proper operational budget. So we have the number of people we need, the facilities we need. There is always room for improvement. You will see some of that, which is the next step. I already told you we operate in Riverside and in Linkov, just to get to orient yourselves. 4 hours driving, 220 miles from Riverside to Linkov. We have the quarantine facility that we're going to visit this afternoon. This building over here is the 1907 Citrus Experiment Station, our greenhouses, and on the main campus we have our molecular biology laboratory and part of the tissue culture operation. This is what we found in 2005, and some of the buildings they gave us, and they were expecting us to serve the industry and be the best program in the world, was some of the original buildings from, I told you the story. And today we're in a much better shape. We have converted, we have get rid of all these old structures. We have a proper greenhouse and old glass house that works okay. But during today's presentation, I added this little note there. For us, allowing a lot of light to come through, or the right amount of light, then we're ended up cooking our plants. So it's very common practice to whitewash our greenhouses, but the moment we do that, we reduce the light. So we haven't managed to find the sweet spot there, and hopefully with all this research and exchange, we may be able to achieve that. Tissue culture, this was our incubator for in vitro work, and wooden box with plexiglass and a fluorescent bulb. And this was our growth chamber, a shade cloth in the greenhouse. Of course, today you will see how we operate, in a modern, small space, but in a modern tissue culture setup. So when we receive budwood from any place around the world, we put it in culture. You see there is no roots here. We put it in artificial media, and within 2 weeks, the budwood flashes. And I know a lot of you have seen this video before, but there is non-citrus people in the audience, so let's do it one more time. So if you were sitting in the microscope performing that tissue suture grafting method that Muresigi developed, this is what you will be seeing or doing. The blade that it's moving, it's the tip of this instrument right there. So that's the blade that does all the movement. And what the person that's performing the STG is doing is removing leaves and leaflets to expose the apical meristem. This last one, which is like a leaf primordium, you cannot see it with a naked eye. So what the person is going to do now is they're going to excise the dome of the apical meristem, which in electron microscopy looks this way, and 2 leaf primordia, which is this and this. These black dots are the smallest known pathogen we spoke earlier, the viral. And you see how they stop right before the apical meristem, because there is no phloem connection between the apical meristem, and the rest of the system. So we are leaving behind the pathogens. That's what all we're doing. Then the white part is a little seedling that we grew in the lab, and there is a triangle incision, and everything you see there, it's research, dozens of years of research, the type of cut, where you make the cut, the shape of the cut, leaving the apical meristem there on the side touching the plant. This thing is so delicate, just breathing on it, you can kill it, you can dehydrate it. So we put the apical meristem inside that little pocket. It stays nice and hydrated. The triangle shape, it keeps pushing down the meristem to be in contact with the cambium layer of the rootstock. And a few days later, that apical meristem starts pushing out. And we recreate a whole citrus variety from those few cells. A few weeks later, we pull the plant out of the tube, and we propagate, we throw away the roots, and we propagate the newly introduced variety into a mature rootstock to take off and create the first mother tree for that variety for the state of California. We put it in growth chambers to keep optimum conditions, and at the end of the day, right there is where the mature rootstock was bent, so we eliminate it. This different color of tissue here is the rootstock that was inside the glass tube, and this stem growing up is the new variety, the lemon, the mandarin, whatever we wanted to introduce. And these are a couple of varieties. Now, when you perform therapy, any kind of therapy, you don't assume it works. You have to, perform tests. So these plants will go in testing. You will see this later. So one project, when it comes to tissue culture, one project we are working on, and you will see it this afternoon, is to actually work, instead of growing especially rootstocks out of seeds, is to grow rootstocks out of cuttings and in tissue culture, so we can avoid all these problems. Some rootstocks, especially new rootstocks, don't produce as much seed. In order to have seed, you have to have outside blocks. You can have seeds only once a year. You have to store them properly, and so on and so on. So this is an interesting project that you will see this afternoon from Dr. Paulina Lamina. So how we can bring innovation to tissue culture, not only for therapy, but for actual nursery production. This grant is funded by the Citrus Nursery Board. So the industry has been growing, as you can see there on the top. When I started as a student, it was less than $1 billion. Now it's 3.6 billion, but it always is, facing challenges, diseases and markets and so on. So the industry has been growing. So the demand for our services has been growing, and we're doing our best to meet the demand. You see how all the graphs for therapy and introductions has been growing as you move to the right, but also the diversity of the material that is needed. It's not anymore a navel and a mandarin. It's all sorts of different material that has come through our program for research and innovation. So have been doing our best to meet the demand on capacity, but meeting the demand on capacity doesn't mean that we are meeting at least our standards and the demand from the industry in efficiency. So when we break down how fast we can push material through our pipeline, we want to be in this green zone you see there, a little bit less than 30 months, 2.5 years if possible. And you can see that we used to exceed that by a lot, but slowly we're pushing it down. And when you take a closer look in some examples here, 104 weeks, that's 2 years. The blue and the orange part is when we work in vitro, so we're doing pretty well there. The moment we start trying to grow that mother tree in our greenhouses, which is the gray zone here, clearly we're losing a lot of critical time, okay? So one thing that we wanted to make innovation is how well we grow that tree from the tissue culture plant on that rootstock, mature rootstock, to bring it to a size that we can perform testing. So we thought, okay, let's maybe build a new greenhouse, a modern greenhouse. You know how well and how efficient the government is when it comes to money. and building things. They wanted like $2 million to build a little greenhouse. It was ridiculous. So that's when we got into researching the different technologies, and we decided on CropBox. And Trip is here today, and he's going to talk to you later. And we said, okay, let's design the prototype shipping container modified for citrus nursery, for citrus plant production. So we have in section A, a tissue culture or rooted cuttings room, and then feed the rest of the structure. You see 3 layers here and 2 layers here as the plant's getting taller and taller, and single layers here. And let's bring light technologies and hydroponics. And that's the time when I heard someone on an interview the other day on TV saying, that's the time that the world broke. So this is the pandemic years. It took forever. Finally, we got the container in March of 22. And you see, and you will see part of the workshop today is actually to visit and walk through and see the lights and the hydroponics on this prototype of shipping container for citrus nursery production. And when you are at Rubid, you will see we're on a hill. They're also stackables. So if this is a success, I'll drop another one on the top. We have an access on the second floor from the back of the hill. So that's definitely something that has drawn our attention. So we can, again, grow plants much, much faster because everybody is waiting for that first mother tree so they can start their production, nursery production, field production. In today's world, competition, companies, patent usually is 20 years. They spend the first 6-7 years of the variety being evaluated, and then it needs another 2-3 years in the nursery, and another 5 years to get the first crop. That's half of the patent duration. I cannot, we, the CCPP, the university, we cannot take 2 and 3, and actually 2 we should, or we can't. If possible, less, but it used to be 5 years or 6 years. We cannot waste 2/3 of the patent before we produce the first 3. So we're trying to compress this as much as we can. Environmental conditions with the MPGU, what we call MPGU, modular plant growth unit, but also what goes on those pots. Until very recently, what was going in those pots is that 1950s potting media, but there is a lot of research out there that you can use organic, and everything back then was sterile, no microorganisms, no nothing. We know today that there is good bacteria out there and good fungi that can help your plant. So this is a project with Dr. Pagliazzi and Sami Ing to use biochar and other components to enrich what we put in this potting media, including, in our case, introducing into the linear economy that we have today in citrus production, principles of circular economy. Take wasted fruit from the ground in the orchards, put it through a fermentation process, in this case, bokashi fermentation process, very old process, and create environmentally microbiologically rich substrates to grow plants faster. And this is like preliminary data. Combining those two things, on the left, trees growing in our greenhouses and trees growing in the MPGU. Like, one of my professors was hating statistics. Why do you need statistics? When you can see, why the hell do you need statistics? He has a point. So yes, we can grow things faster and better by combining all these technologies. The second part that we were losing time is in diagnostics. You see the yellow part there pushes us over the 2 years. So we have been investing a lot of time on improving diagnostics, which has been a very common process in the CCPP, from visual symptoms to biological indexing to serological techniques, today doing high throughput sequencing and so on. So one project that my student Stacy Comstock that you're going to see today, based on literature, was like, if you expose plants that they have been inoculated with a disease and you want to see symptoms, if you expose them to different wavelengths of light, you may see differences. And indeed we did. The first and most important thing we saw is that the healthy plants look healthy. They don't become chlorotic, they don't develop spots and so on. And when we introduce a pathogen, you actually see beautiful symptoms on those plant indicators. You see these yellow lines here and those yellow spots. And when Stacy started analyzing more and more data, these are complex graphs, but what you want is heavy symptoms, so which is to the right, as fast as you can, which is low on the axis or on the y-axis, and the size of the bubble is how much virus the plant is building up. We haven't found a correlation on that yet, but you can see that different viruses, when exposed in primarily majority of red light, 95% red light, 5% blue. 65, it looks like over there, right? 95, you see stronger symptoms quicker. So in our greenhouses, in order to give a clean bill of health to a new introduction, we inoculate plant indicators from the lemons, with tissue from the lemon, the mandarin has been introduced, and then we inoculate indicators with known diseases. When the known diseases show us symptoms, and the new introduced variety doesn't show us symptoms, it means that the newly introduced variety doesn't have a disease, because if it had a disease, it would have shown symptoms when the positive control shows symptoms. So the sooner we see results in our positive control, the sooner we can say the new introduction doesn't have the disease. And you will see this afternoon in our greenhouses. And we continue the classic work with all the molecular tests, including, as I told you, high throughput sequencing in collaboration with Oklahoma State. They have a beautiful facility and program there. When we bring the two together, the biological tests and the laboratory tests, then I write a letter to the federal and state government, and we told them, you remember, you gave me a permit because it was illegal to bring citrus into the United States. Now I have those tests, please allow us to release the material into the environment. And they do. And with all these innovations, we have managed, you see all these spots splatter all over. This is how many months it used to make materials coming through the program. Now we have been compressing our introduction towards those 30 months time frame. I got the signal, we need to move faster. I think we're done because now we're moving to the release. I want to show you how the foundation block used to be. It used to be outdoors, 98 the first greenhouse, 99 expanded, 2010 duplication, 11 building a positive pressure greenhouse with two sections to preserve all our material. This is the time, you see, early 2000s, where the HLB epidemic was spreading in Florida and threatening California. So we had to build layers in our system, how to protect our varieties. This is how the foundation block looks inside the protected foundation block, greenhouse on the greenhouse on the right. One great innovation is that we have digitalized the whole budwood distribution process. Everything is scanned, nobody's writing down anything, and of course we have avoiding mistakes and so on. This is something very scary that happened in 2012. The first HLB tree in California had 23 unknown grafts on it, which meant that there were people out there making trees outside the official pipeline we have been discussing all this time. So we open our system to everybody in the state of California. And last time I checked, in 2024, this is 2023, there were over 12,000 people ordering clean budwood from us. So there was 12,000 people out there all this time getting their budwood, who knows where, making trees for their backyard. So that was a very, very important, and you can see the green lines there, how few commercial growers there are. 108 commercial growers versus 9,500 people out there getting budwood. Another innovation, recent innovation, is that we started this automatic email. The nurseries in California, just before a variety is ready to come out, they get an email, are you interested in this variety? And they let us know how much interest they have. And that way we can get prepared. Right now, when a variety is released from quarantine, we have one tree. But that's when the demand for budwood is the highest. So the graph is like this. We don't have enough budwood when the demand is higher, and as the demand goes down, we get a lot of budwood. So by knowing in advance how much budwood is needed, we are targeting to give nurseries upfront as much budwood as possible. This is innovations we perform in high throughput sample testing. And I'm not going to show you this video, but essentially there is a piece of equipment that now our collaborator with TESS, with Technology Evolving Solutions, has a grant to bring it to the nursery testing program. And what this technology is going to do, has a grant with the Citrus Nursery Board, instead of testing individual mother trees, we can combine them and pull them. It costs $170, $180 per mother tree to the nursery to test it. So by combining, we hope to bring down the cost. And why the time is perfect now? In the early days of the nursery testing program, these red bars correspond to 500 samples over there. The program could test 500 samples a year. As a result, there was always this black line represent viroid infestation in the nurseries because they're very easily transmitted with cutting tools, as we spoke. When we set up the mandatory program and the new technologies for testing, we can test thousands of samples a year. And you can see, and that's a beautiful graph also in general, when you test fast enough, it was a big conversation during the COVID pandemic, you can beat the infection, right? And we brought it to zero down here a few years ago, and we just had a little blip. We found 2 new trees in a nursery that had a viroid infection. And I put a blue arrow there to remind us that based on our conversation with those nurseries, this could be very well a blitz cleaning tools issue. People keep forgetting, so please remember and remind. So it's a systems approach. None of the players in the pipeline can solve the problem on their own. We provide the foundational material, but it's the leadership and entrepreneurship and vision of the industry that makes this thing work by supporting, all these different programs that we have in California. So it's time for our break. If you want to ask something.